Hello, assalamualaikum everyone. I hope you're all keeping well and keeping sane and keeping healthy, and of course abiding by the rules that's set during this PKP or MCO movement control order. So obviously there's a very good reason why we cannot continue lectures as we normally would, because there is a very contagious virus that's spreading everywhere, and we're going to cover a bit about that particular virus in this course. And I think it's a very, not to say nice, but it's always very humbling when you can kind of match what you're learning in theory with what you're able to observe happening in real life, including your own life right now. But there are some severe implications of the lockdown and also the virus spreading. Although Alhamdulillah, I think Malaysia seems to be in a better place than some countries. We still want to be very cautious, and so even though the MCO is due to be lifted by the 28th of April, subject to the next announcements, there's a chance that we will be going, not a chance, it's almost certain that we will be continuing most of our lectures remotely. So I'll just be pre-recording them and uploading them online, and you can view them at your own preferred time. Of course, it's better if you can keep up with the lectures as they come up, because I will also do some quizzes every week just to make sure everybody is on board. Yeah, by the time we've covered the entire syllabus, I think it will entirely all be online. So there is some necessary revision to the way that we're assessing you as well. So this table right in front of you is basically, I hope, what you're already aware of, because we've put this on eLearn, but I'll just talk you through what we're planning to do at this stage. So although we're going to have our lectures entirely online, if we are able to arrange for it, we would like to have at least one practical session, and that would be the diagnostic practical that is usually the second half of the immunology experiments. So the first half is usually the animal immunizations. That's not able to be done this semester for very good reasons. But the diagnostic practical might still be able to be done. We'll just arrange for you to come in smaller groups, and then you can run the tests and get your data. But if that's also not possible, then there are ways around it. We'll give you some data, and then you can understand the data, analyze it, interpret it, and then write a report, and still understand how it would work if you had some data from a diagnostic assessment like an ELISA, for example. So that's the lab report. So obviously we've shrunk down a little bit of that component to 10%. And we're replacing some of that with some assignments to be determined. So the first assignment, or 10% of the assignment, will be covering part 1 under Dr. Yahya, and then the other 10% will be from me, and I'll devise a group assignment that you can do together, probably a written assignment like an essay. So we also have a larger weightage for tests. So we have one test dedicated to part 1, and then another test dedicated to part 2, and then we'll have a third test where we'll combine both part 1 and part 2, so roughly 5% from each part. And then, because I love presentations, and in fact this class has given me some of the most creative presentations. And what I usually do is I ask people to come up with a video, like a 5-minute video that explains an immunological concept that was covered in the course, or maybe a little bit beyond the course, that anybody from your grandparents to some school kids can actually appreciate and understand. So that's always been really fun and creative, and usually we do that by group. And quite conveniently, because we are remotely doing this, using video is quite convenient as well. So I'll give more details on that closer to the date when we will start looking into the presentation assignment. Then we have 10% for quiz. Originally this was 5%, but now we'll just add another 5% for that. So I think you've already gone through a quiz with Dr. Yaya, so that makes about 5% of your marks. And then for part 2, just to keep you engaged every week, what I'll do is I'll open a quiz covering 2 lectures every week. So that should amount to roughly 4 quizzes. I think we have about 5 weeks. And then the best out of these quizzes, it will make up your 5%. So for now, I'm thinking I want to be nice, because I know everybody is trying to survive and adapt to the situation. So I want to give you the best shot that you have, but I do hope and I expect everybody to attempt all 4 quizzes. So it's basically the best out of 4, not take it once and then if you get 5 you don't have to take the others. That's not how it works. Take it every week and I will take the best out of the quizzes that you've attempted, okay? But it's important to attempt the quizzes just for your own learning as well. And finally, although not a lot of courses have decided to keep up with final exams, we think it's quite important because the final exams is going to allow you to reach some level, higher levels of thinking when it comes to synthesizing and understanding the materials in the course. So what we want to do is do an open book exam where we'll open it up, give you the questions, give you about a day or 2 to actually prepare your answers, and you can then upload it back on eLearn. And the questions will be 10% from the first part and then 10% from my part, this is the second part. And altogether this adds up to 100%. And just for fun, I will try to come up with some online activities through eLearn, like a forum, just to stimulate some discussion and interest. And you know, a chance for us to interact in the class, because otherwise it really is just, you know, me talking to my laptop and hoping that you're listening to me wherever you are, if you're able to. So at least we can have some interaction there. Right, so let's get into it. Of course, if you have any questions, you can ask me now, but you can email me. My email's right there. Also, feel free to comment after the lecture in the comment box on Loom. So any way you can get my attention, if you have a question, I will try to answer them as soon as I can. Right, so let's get into it. So this is part 2 of BMT 311 Immunology, and we'll start with a topic very near and dear to my heart, which is immunological memory and vaccination. So the learning outcomes for this part, or rather this lecture, is just that you'll be able to describe the immunological response and memory against infections and vaccination. And you can explain the difference in how the immune system processes different types of vaccines, okay? And our main distinction here are live, and then live vaccines and everything else. So not just attenuated, but basically if it's live or non-live vaccines, yeah? Right, so this is basically the schedule that we have today, or this week. It will be the immunological memory and the immune response to infections. And then you can have a look at the other topics coming up, like the immunodeficiency and HIV/AIDS, immunological tolerance, allergy and hypersensitivities, autoimmune disease, cancer immunology. And then we start going into real applications like diagnostics and detection methods. And then we go into therapeutics and organ transplantation. And then we'll have one big test, that's the 10% that I talked to you about. And then because it's going to be Raya and Eid break, not sure what we will be doing during that time. It's so much uncertainty. But it will still be something that we can celebrate. A lot of things for us to be thankful for, really. And then finally, we'll do a revision of the materials, and I'll go into a bit more detail about how to approach an open book final exam. it's not going to be super different from your final exams that you have to do in the hall, but the level of difficulty will be a bit higher because you're allowed to go and open a book and confirm whether or not you understood or your answer has the right facts. But the key is to understand the question and understand what the question's asking. So I'll go through some past year questions, and then hopefully that will give you some idea what it is that we're asking when we ask certain questions. And it's not about telling us everything you know, but it's about answering the question. So that still applies even if it's open book and on eLearn. Okay? Right, so going into today's lecture about immunological memory and vaccination, the first thing would be to recall what is the immune system. So I've also put up a video or a link to a very nice simplified summary of the immune system on YouTube by Kutzgesagt. I don't know, I never know if I pronounced that right. So that should give you a bit of a refresher, but here's also a little bit of a refresher. So we remember that immunity is basically meaning you are exempt. So exempt meaning you are not going to get sick, basically, if you have immunity to something. But of course, it's not as simple as that. But what the immune system, what immunity now has become, or how we understand the immunity and immune system, is that it's actually a collection of cells, tissues, and molecules that protects the body from numerous pathogenic microbes and toxins, and actually not just that. So there's also immunity in chronic diseases as well. So they play a much broader role than we ever thought they did before. But for the purpose of the topic of vaccines, we're talking about their main function, which is to prevent or limit infections through a concerted immune response. So the key word here is concerted. So they work together to ensure that you are protected while you have some invaders inside you. So we divide this into innate immunity and acquired immunity. So all of this should be fairly familiar to you. And these are some of the things that can invade you. So we've got viruses, toxins, carcinogens. And then also there are some food antigens, actually. So when we talk about allergies, that's because obviously we have a certain inappropriate immune response to things that should technically not harm us, but also part of the immune system. But less so relevant for vaccination, which is the topic of today's lecture. So let's recall also adaptive immunity, because that's very, very crucial when we talk about vaccination. So the adaptive immunity is basically our second line of defense, and it's activated after the innate response has first been activated. So this involves the activity of these very special immune cells called lymphocytes. So the main ones we talk about are the B lymphocytes and the T lymphocytes. And what they do is they basically remember. They remember anything that's invaded, and then they'll react much more aggressively and faster the next time they encounter the same organism. And it's specifically through memory B cells that we have what we call this recognition, or specific recognition of a second time intruder. So what are some of the attributes of the adaptive immunity? Okay, so this is also something you should be familiar with already. It should actually not attack against normal host body cells. It should not. But of course, this is all, this is the black and white version. The reality is a bit more complicated, but by and large, it does not attack our own cells. It's also very specific. So it technically would act only against one particular molecular shape and not against the other. So the molecular shape we're talking about are the antigens. So whatever proteins or whatever molecules that they were produced against, that's what they're going to recognize, and they're not going to recognize anything else. Although there are some instances of what we call cross-reactivity, especially if there's some similarity in shapes, so that might happen. But by and large, they're quite specific. They're also inducible, so they activate only in response to specific pathogens, and they also have clonality. So once they've also been activated, once they've found something that they recognize that they can bind to, they'll make multiple copies of themselves, so that there'll be others out there who can detect and recognize that same antigen. So they form an army of clones. So if you remember the Star Wars movie, Clone Wars, or is it Star Wars? I think so. So in terms, the other attribute is memory. So we already talked about this. This is the ability to adapt and respond faster and more effectively when you encounter the same thing later on. So again, this is still just another refresher. We've got basically your humoral immunity and your cell-mediated immunity. Humoral is usually what's driven by the B lymphocytes, the B cells, and cell-mediated is what's driven by the T cells or the T lymphocytes. We'll go into this in a bit more in the next section when we talk about how these work for different infections. So when we talk about pathogens, right? We talk about pathogens. We can talk about viruses. We can talk about bacteria. We can talk about fungi. We can talk about parasites. And these are some of the, and all of these we kind of categorize under infectious disease. And so these are some common agents of infectious disease that attacks different organ systems. So we have here influenza, measles, Epstein-Barr virus, Streptococcus pyogenes, and of course our very famous friend coronavirus right now. So that also attacks the respiratory system. Then you have things like the hepatitis viruses that attack the liver. You have enteroviruses, rotavirus, cholera, salmonella. Those are all for the gut. And then you have TB, that's in the what we call the lower respiratory infections. So these are the upper respiratory infections, lower respiratory infections. And some of these actually would kind of overlap, although you have one that's more dedicated to attack one area. Same thing with so basically all of these are some general categorizations, but be aware that pathogens don't really care what system you have. Like, they'll bind to whatever and they'll go to wherever they can, depending on how they interact with the cells present in those systems. Then finally, for the sexual transmitted diseases, you've got chlamydia, syphilis, and the human papilloma virus, which some of you may know is also a cause for cervical cancer. Right, so the question is, how can the immune system recognize, remember, and protect against so many different things? And part of that is this idea of antigenic specificity and antibody diversity. And I think I'm not going to go into a lot of detail how this is generated. I think this was covered in part 1. But it's basically there's a multitude of patterns that can be recognized by the receptors on the T cells as well as the receptors on the B cells, which later make up your antibodies. So through a bunch of recombinations on the genetic level, and then also during activation when you actually encounter the antigens, that's how they're able to recognize different things. But the key thing is, once they're actually able to recognize, there is clonal selection and expansion. So you know that before even any pathogen invades, the B cell precursors are already rearranging the variable parts of the antibody genes to make sure that you've got as many possible options as that can bind an antigen. But when an antigen actually comes in and binds to a clone with a specific membrane antibody, that's when the B cells, which are now antigen specific, are activated and they can secrete these antibodies. So the antibodies is what actually, which were formerly the receptors on the B cells that bound to the antigen. So that allows these B cells to actually produce a lot of clones, which then rearrange the DNA, which then creates the gene coding for that specific antibody. And after that exposure has ended, the plasma cells, which is what was producing the antibodies, will start dying. But you'll have the other differentiated B cells called memory cells, which still remain in much larger numbers, and they're just waiting for the next time that antigen comes again, and then they'll start expanding and become and dividing into plasma cells to make more antibodies as well. So this is basically a schematic of how that works. You've got the stem cells, and then the deletion of the self-recognizing cells, and then you've got the different cells that undergo clonal selection and expansion. Once it actually meets the antigen, that it has a pattern that can recognize the antigen, and then it makes multiple copies of itself. So yeah, so basically that's it. You've got different possible combinations. The self-reacting patterns are deleted or die. And then the patterns that encounter an actual antigen that match are selected and then made multiple copies of. Yeah, and that's basically the whole idea of immunological memory. That we have these very, very cool cells called memory cells that can actually remain for decades, even after you're done. You know, like you've definitely forgotten you ever got sick from that bug, but the memory cells remember, even though there aren't any antigens present to stimulate them. They've already retained that ability to recognize that antigen. So that's why when for some reason you encounter it again in the environment, you definitely won't get as sick as you did before, typically. This is the whole basis of why immunological memory gives you immunity, gives you that ability to be exempt from getting sick again from the same thing, in theory. Yeah. And this is basically how that happens. The first encounter with the foreign antigen gives you a primary response, which we just talked about just now, where you actually activate the B cells. The B cells are then clonally selected and then cloned and then expanded, and then they deal with that infection and try to get rid of it. Okay, job done. Infection is over. Bug has been eliminated. Memory B cells stay on. Okay, memory B cells stay on, and they are the ones that will then recognize if a pathogen or an antigen has actually been in before. It's like, wait a minute, I know you. Okay, I know how to get rid of you. So they'll basically mount a secondary response, which is, as you can see in the graph, is much stronger compared to the primary response, even though it's been quite a while. And this is basically data from an animal. What happens when you first introduce the antigen? You'll get a primary response, and then you wait a while, give it again, and then the response is much higher. But notice, in contrast, the lighter shades of blue, which is the innate response, it's actually the same. It's the same shape and the same magnitude. So the innate response does not remember a pathogen. It's very general. It will just keep on behaving the same way it would, regardless of what it meets. Whereas the adaptive response is more, because it's specific, it actually knows, oh wait, I've seen you before, so I'm going to make sure all out, get rid of you. Whereas the innate's like, same old, and not just another invader. So it's the same magnitude and type of response. And so understanding that we can actually get memory and get immunity, of course humans are very industrious, we then try to use this so that people don't even need to get sick, and they can still be protected to some degree. And so that's the goal of active immunization, is to trigger the adaptive immune response in a way that will get that protection, without actually having a person have to go through an actual infection. So if the immunization is successful, once a person encounters the actual pathogen or the actual bacteria or virus, they will automatically elicit a secondary immune response that is much stronger than a primary response, which they would have had made when they were vaccinated earlier. So active immunization is usually done when you actually get infected, but also through a vaccine. So that's how it works. And you've got different types, different distinctions. I mean, usually when we say vaccination or immunization, we are talking about active immunization using either a live or a non-live vaccine. But immunization can also mean different things to different people. You can say passive immunization, where you actually just transfer antibodies, and that's naturally done, when you do things like breastfeed your child. So that's a very good way to ensure the immune system of a child that's naturally very, very weak is supplemented with some help from antibodies from the mom. Then, of course, here they're calling active immunization naturally acquired, when somebody actually gets the infection and then develops immunity to it. But this is what we usually mean when we say vaccination or immunization. It's when we actually give the antigens or even the attenuated live vaccine, and then we stimulate the immune response. But there's also passive immunization that's artificially given, where you kind of inject just the preformed antibodies without requiring to stimulate the immune response. So that also helps quite a lot for certain infections. So, in fact, you can read up on this, but that's one of the ways that they're trying to see if they can treat COVID-19 as well. Just kind of giving plasma from recovered patients, because they contain the antibodies that can also help current patients fight off the infection. Right. So let's go into a little bit of a history of vaccination. It's a brilliant idea. So who actually came up with it? As far as we know, the first vaccination occurred in 1798, where Edward Jenner observed that milkmaids, so you know the milkmaids, the ones who the milk from the cows, before we had all the factories and all the machines. So we had milkmaids. So they were obviously with cows a lot, and they would develop what was called cowpox. So they'll have some blisters on their hands. But this is a fairly mild disease compared to another pox, which naturally infects humans, called smallpox. So smallpox is horrible, kills a lot of people, very miserable. And he noticed that the milkmaids, although they had cowpox, they didn't get smallpox. So he wondered, or he reasoned, that maybe if he could take some of whatever's in that cowpox pustule or the blisters and then put it on other people, they will sort of have the same protection that the milkmaids would have, and they wouldn't get smallpox. So to test this idea, he actually took a little boy, I think his name is James Tibbs, and he inoculated, which is kind of like you scar or this, I don't think they inject it, but he basically just gave some of that fluid to the boy through inoculation, and then infected the child with smallpox. Now, nowadays this is almost impossible to do. It's very difficult to test anything children. It's very important though, and he's very lucky that the boy actually survived, but at least this is the boy that we know survived. So this boy survived, and he didn't get sick from smallpox. Okay, and that basically gave us the first vaccine against smallpox, and this is called, at the time they called it variolation, because the virus that caused the cowpox is called variola. And now what you can see here, since that concept became further developed for other diseases, the difference is very, very vast. So before we had a vaccine for all these diseases, you had so many pre-vaccine cases. And then post-vaccine, some of them dropped to zero, and some of them dropped significantly. But you still have certain diseases like pertussis or whooping cough that has reduced cases, but it's still around. So it's also a reflection of how good the vaccine is, or how the immune system works against the pathogen. So there are basically 5 main types of vaccines, and when we talk about how good a vaccine is, the type is also an indication of how it's going to perform. So we have toxoids, inactivated bacterial components, for example, diphtheria, tetanus, and acellular pertussis. We also have inactivated viruses. So these are whole viruses, but then they've been inactivated, which means they can't replicate. So we have these vaccines for influenza, hepatitis A, polio, Japanese encephalitis, And we have conjugated bacterial polysaccharides. So these are basically the polysaccharides of the bacteria that's been linked to something else. And so they're not the whole bacteria, definitely not alive, cannot replicate. And some examples are the Haemophilus influenza type B vaccine and the pneumococcus vaccine. And usually what they do is they try to include different strains of the bacteria in one. So it gives protection to different kinds of the bacteria. So you know, certain, for example, even influenza, you've got flu A, flu B, and then you've got different strains. So they'll make combinations so that you're protected from as many strains as relevant for that particular disease. And then you have the live attenuated ones. So the live attenuated ones are special from the others because they're actually still alive. That means they can still replicate, but they're weaker. So they've been made weak through usually serial passaging, which means as you keep growing it over time in the lab, you'll get a weaker and weaker version. But it doesn't mean they cannot still cause some illness, especially in some people. But most of the time, they are much, much easier for the immune system to get rid of. We also have some other types of vaccines that are still under investigation, like DNA vaccines and vector vaccines, but none that I know are licensed yet. So the rule of thumb, when we talk about types of vaccines, live vaccines, the ones that can replicate, gives you the most protection. Why? Because they're the things that's closest to what would actually make you sick. But obviously they have the risk of actually making you sick. So it also can't be given to people who have any immune deficiencies or are immunocompromised, because then they would actually get sick from the vaccine. Now, inactivated or subunit vaccines are less protective because they're a bit more different from what would actually make you sick. But because of that, they don't often make you sick after you get vaccinated, so there's less risk. And they usually need some booster doses because they're just given, it's mostly the antigens rather than the replicating organism. So if you think about it, the whole idea of vaccination is you kind of want to train your army as well as you can so that they're prepared if the war ever comes. But you don't want the training to be so rough and so terrible that you kill the army before the battle even starts, right? So there's a bit of a balance and a trade-off there. So these are some more details of different types of vaccines, and also their examples of the diseases that they protect against, their advantages and disadvantages. So you can have a look at that. So now we're going to go into how the different vaccines actually stimulate the immune system. And here we're going to talk about the role of the MHC, or the major histocompatibility complex, which I hope is very familiar to you now. So one key thing is that B cells can recognize an antigen without the presentation through the MHC. T cells require that an antigen is displayed through the MHC. So the MHC molecules are sort of like the cup that holds the antigen, whereas the B cells can just go and can get the antigen without anything holding it. And there are two main classes of the MHC molecules that we're going to talk about in this lecture, which is the class 1 and the class 2. And they differ slightly in how they're arranged on the quaternary level. And they differ in the cells that express them and in the source of the antigens that they can present to the T cells. So class 1 molecules are usually expressed on nucleated cells, the cells with nucleus, and class 2 molecules are usually for the B cells, macrophages, and the dendritic cells. So this is some differences between the class 1 and the class 2. If you can look at this here. So some of it similar, and then you've got this major difference in that beta 2 microglobulin quarter for the class 1. Now, what happens is that the class 1 molecules are usually the ones that process endogenous antigens, and they'll present it to CD8 T cells, whereas class 2 molecules will process exogenous antigens and present it to CD4 helper T cells. So there's some implications to this. Well, I'll go into that in the next level. So the type of vaccine will actually be different types of vaccines will be processed differently. So if you're getting vaccinated with a subunit vaccine or a killed vaccine, this will largely produce an MHC class 2 restricted response, which means you won't get much response from your CD8 cytotoxic T cells. But if you get vaccinated with a live vaccine, it will mimic natural infection, and both the MHC class 1 and the MHC class 2 response will be produced. So what this means is, if you get vaccinated in general, with any vaccine, you will get an antibody response. There will be antibodies made against whatever vaccine you were given. But only live vaccines or these experimental DNA vaccines can get you a cytotoxic T cell response. All right? So how does the vaccine response start? Well, first it starts with the introduction of the vaccine, most often through an injection. Of course, there are oral vaccines as well, or even nasal spray vaccines. But let's say for this purpose, we start with an injection. So with the injection, the vaccine is introduced to the body. You have pathogen-associated patterns in the vaccine attracting antigen-presenting cells, such as dendritic cells and monocytes and neutrophils that are just generally around looking for any signs of danger or trouble, yeah? So, oops, just moving my face a little bit. Yeah. So the vaccine antigens and whatever adjuvants that's present in the vaccines, we'll talk about what adjuvants are a little bit, but basically together they're just sending out danger signals and activating the monocytes and dendritic cells. So the monocytes and dendritic cells then alter their surface receptors, and then they'll start migrating along the lymphatic vessels. They'll go to the lymph nodes where they'll meet the T and B cells, and then they will stimulate those T cells and B cells. Now, again, what I mentioned earlier, when you have a non-live vaccine, it's only going to be processed through the MHC class 2. So the only T cells that will be stimulated are the helper T cells. So helper T cells are really important because they're the ones that help drive the B cell activation and differentiate them into plasma cells or memory B cells. So once that happens, the plasma cells will start making antibodies, and the memory B cells will be maintained for a secondary response. Okay? So this is the MHC class 2 presentation. You've got the extracellular self or foreign protein being endocytosed, and then it will be processed through the MHC class 2. Then the protein will be degraded, and then it will be carried forward and presented through the MHC class 2, and it will start stimulating and activating the T helper CD4 T cells. Now, this will give you what is called the humoral immune response to a vaccine, and this is what happens for all vaccines. All vaccines will produce a humoral immune response. Yeah, first we'll have some low affinity IgM antibodies, and then later you'll have IgG antibodies that have higher affinity. So this is called isotype switching. So if you recall, antibodies have different isotypes or classes, and they also have different subclasses as well. And what kind is produced by a B cell depends on some T cell cooperation, and it's called switching. So first it will make IgM. This is usually what we call it during the immature phase, and then once it's more mature, it will start producing IgG, and it's a later response. And you have your antibodies usually reach the peak at about 2 to 6 weeks after infection, and then it gradually falls. So that would be the primary response. But then once you have the secondary, so basically this is the secondary response. So you can notice that the IgM is produced around day 3 in a primary response, and then the IgG is produced a bit later. But in a secondary response, the IgM is produced much faster, and the IgG is actually produced around the same time as well. And then it will stay on while the IgM will disappear about a week after that. Now the difference is with live vaccines is on top of the MHC class 2 presentation, you also have the MHC class 1. Now why is this? Because live vaccines are able to replicate, and it's able to spread. So it's not just, you don't just inject it and then it just stays there and it has to be picked up. It actually has the ability to go around somewhere else. This means it will send out even more danger signals. It will produce a greater innate response, trigger more pathogen recognition pattern receptors, and it actually stays a while because it's also making more copies of itself. So that means there's more antigens that are going to be present. So in general, you'll have a higher antibody response, but also on top of that, because it's going to be processed through the MHC class 1, it will be engulfed by the APCs first, and it will replicate intracellularly, be processed as endogenous antigen for MHC class 1 presentation, and then it will stimulate your cytotoxic T cells. So overall, you'll have much greater immune stimulation from a live vaccine. But as we talked about earlier, there's also the risk of having something that is so close to its natural form that it might just get you sick. So in contrast to what we saw with the MHC class 2 presentation, where you originally had an extracellular antigen, so this one, for example, this live attenuated virus, it actually binds to the receptor and gets endocytosed. So here it's actually being processed. It's actually making multiple copies of itself and all its genetic material. And all of these antigens will now be processed and presented on the cell through the MHC class 1 molecule, so HLA or MHC class 1 molecule. And that's the one that's going to stimulate your cytotoxic T cells. Right, so that's just the vaccine itself, right? Whether or not you're using a vaccine that's still alive or a vaccine that's not alive at all, and can be different versions. Additionally, inside a vaccine, you also have what's called adjuvants. So these adjuvants are not anything related to the infectious agent, but they're general materials that we know can stimulate some form of inflammation or increase the immune response. So they can be mineral salts like aluminum or calcium, or they can be oil and water emulsions like the Freud's adjuvant. Like they can be synthetic toxins from bacteria, saponins, and you also have things like LPS, lipopolysaccharides, that comes from the gram-negative bacteria, or flagellin. So different parts of unrelated microorganisms that we know can stimulate pattern recognition receptors, even though they're not directly related to the vaccine agent. They'll be used just to stimulate that immune response for the innate immunity arm. Okay, so that's adjuvants. The other thing about vaccines is this idea of boosters. So we talk about the vaccination as if it's one injection and then it's done, right? For some vaccines, that's true. You only need one round of vaccination and then you're good. So an example of a vaccine that is only given once is the BCG vaccine, which protects you against tuberculosis. So the BCG vaccine is only given at birth, and then it should protect you throughout your childhood. There are no boosters that are known to increase the immune response, so we don't give boosters. But some vaccines we know will only work if you keep giving supplemental doses of the vaccine. So this can be, for example, the DPT or the DTP vaccine. So it's actually a series of 4 shots during a child's infancy. And you also give another booster once they reach school age, so when they're in first grade, or if they were 4 years old. So what this does is basically the second or third episode of exposure to the antigen will drive new rounds of secondary responses, and it kind of hones the response and ensures higher levels of protection against the pathogen. So that's booster. So the other concept relevant to vaccination is this idea of cross-protection. So even though you have vaccines that are made to protect against specific diseases, we've also observed for some vaccines, they are able to produce cross-protection. So they don't just protect against the disease that they're meant to protect against, they can also protect against some other diseases that have some similarities, especially if they have some similar antigens and things like that. So, for example, you have the flu vaccine, the influenza vaccine. So that might be a formulation that is going to protect you against whatever strains they've included in that vaccine, but it might also protect you against some other strains that are similar. Then you've got the HPV vaccine that's also made against a group of strains of HPV viruses, but it can also protect against some other strains of HPV. Now, BCG vaccine, so that's really interesting. It doesn't just protect against TB and some other mycobacterial infections. Mycobacteria are a huge group of bacteria, but it also reduces some other respiratory bacterial infections. So there's some good data on that about the BCG vaccine cross protection. And I mean, you can read up on this. I'm quite cautious about the interpretation and the way they did the study, but there are some researchers who are noticing that countries that have BCG vaccination versus countries that don't have BCG vaccination, they're actually having different levels of death from COVID-19. Mainly the ones that have general BCG vaccination in the country have less COVID-19 mortality compared to countries that don't have the universal BCG vaccination. The data to me is not very convincing, but it's an interesting idea because BCG we do know it protects against a lot of respiratory infections, only that it's usually other bacterial infections, and the COVID-19 is caused by a virus. So something to think about. The other thing about vaccines is waning immunity. So waning immunity means you lose whatever immunity that the vaccine induced after some time. We don't understand why this happens, and it's a bit of a pain, obviously, because you would hope once you've been vaccinated, you would be protected forever. But that's not what happens with quite a few vaccines, actually, notably the pertussis one. That's why you still have a lot of cases of pertussis, even though the vaccine is given 4 times. And the vaccine is usually has high coverage, means a lot of people get vaccinated for pertussis as part of the DTP vaccine. It's another example is also rotavirus. So I talked about pertussis just now. So rotavirus is another example where you don't have as much protection, but it's still often that you will protect people more than not vaccinating them for these diseases. Now, there are also diseases that don't have any vaccines. It's not that people haven't tried to make vaccines, but there are different reasons why we cannot get a good vaccine to work. Sometimes it's because the organism itself has just so many different variants, molecular variants, and we don't know what is the most important one to target to get protection. Or some diseases require a vaccination regimen that's so complex to generate protective immunity that it becomes not feasible to actually implement it on a wide scale. Or for some reason, the virus or the bacteria that causes the disease, even in nature, does not actually produce protective immunity. So that makes it quite hard to use a vaccine to mimic a natural infection if even the natural infection wouldn't give you that protection, which means you could keep getting it again and again. So there's some examples of diseases that don't have vaccines. So HIV/AIDS, one of them, hepatitis C, and malaria and dengue. So I put an asterisk there because you might find in the literature that there are some vaccines available, but they're mainly limited in limited populations, and they don't work the same way other vaccines work. For example, the malaria vaccine is actually not a vaccine to protect the individual from getting malaria. It protects the individual from actually spreading the malaria. So it works a little bit differently. And of course, we don't have a vaccine for COVID-19, which is something that we really, really want and we really, really need if we want to get back to normal life. And I'm going to put this as a forum question, and maybe some of you can share some ideas what you think will be the main challenges in getting a good COVID-19 vaccine as soon as possible. Right, so that's the end of this lecture. Let's have a quick recap. So the immunological diversity and memory are important features of the adaptive immune system, and that's what allows us to use vaccines to prevent disease if we ever encounter the same pathogen. And there are different types of vaccines, but the main distinction we make is the live and non-live vaccines, because they're treated differently by our immune system. Mainly that the live vaccines will be processed by both the MHC class 1 and the MHC class 2, and so that will give us a cytotoxic T cell response as well as an antibody response, whereas non-live vaccines will only be processed through MHC class 2, and they'll only produce antibodies, but they're also usually less risky, better tolerated, and as a fact they also need to use boosters. So this is a cute cartoon about vaccines, and I think that people have very strong opinion about vaccines, or really, really pro it, or they're really, really against it, and some people just not sure, but they'll go along with whatever the government tells them. Sometimes not a bad idea, especially when it comes to public health. But the reality is that I think when it comes to outbreaks like this COVID-19, all those questions about are vaccines safe or is it unnatural, I think they're a lot less quieter now, because we realize, when it comes to infectious disease, we kind of want to get as much help as we can, and that includes using vaccines. That it's one of those things where you realize how important something is now that we're all facing some amount of trouble from this new coronavirus. Okay, so that's the end of this lecture. Some references here. Feel free to have any questions, and I do hope you will put in some thought in the forum discussion. It's not compulsory, just for fun. All right, see you in the next lecture.